yesterday we had discussed about the cloning vectors and plasmid we had discussed. So today one topic was left before it, that is of separation and isolation of DNA fragment. Okay, so I'll tell you it with the help of gel electrophoresis. This technique is gel electrophoresis, means हम isolated DNA के fragment को अलग कैसे करते हैं. Okay, suppose this is the gene of interest. Okay, in this part you required. Okay, this is part. Okay, suppose it's responsible for insulin. DNA में ये हिस्सा इंसुलिन के लिए रिस्पॉन्सिबल है. सो विद द हेल्प ऑफ रेस्ट्रिक्शन एंडोन्यूक्लियस you can cut DNA and make it DNA fragments, and these fragments, the cutting of DNA by restriction endonuclease result in the fragment of DNA. Okay, ये सारे fragments बन गए, like it is like this. Okay, and you want to separate them. Okay, DNA fragments are negatively charged, student, as I told you earlier also when we had read DNA. So you know that DNA is negatively charged, or they can separate towards anode. Okay, under an electric field. You can see here electrode is there. Electrode जहाँ पर होता है बेटा, it generate cathode and anode, two parts. Okay, so here you can understand that the most commonly used matrix is agarose. So inside of it which gel is present, agarose gel is present. Okay, एक जेल है बेटा इसके अंदर, that is known as the agarose gel. That's why it's known as the agarose gel electrophoresis. Okay, so this is a natural polymer and which is extracted from seaweeds. So DNA fragments separates according to their size through this area. Okay, if we put all these fragments in it, the smaller the fragment size move fastly. Are you getting my point? जो छोटे होते हैं वो तेज तेज भाग के पहुंच जाते हैं. Clear? The smaller fragments move fastly as comparative to big one. Okay, so you can separate DNA fragments with the help of this method. Okay, so one staining medium we can use here, that is known as the ethidium bromide, which has given in your NCERT book, ethidium bromide. Children, if you feel any problem in it, in live class again I will explain all the things. Okay, so ethidium bromide, which is followed by UV radiation. Okay, because we know that DNA fragments can't be visible विदाउट स्टेनिंग. बिना उनको स्टेन करे आप उनको नहीं देख सकते. ओके, सो फॉर स्टेनिंग वी आर यूजिंग इथीडियम ब्रोमाइड. प्लीज नोट डाउन दिस थिंग. इथीडियम ब्रोमाइड इज वेरी इंपॉर्टेंट, बहुत बार पूछा जाता है एग्जाम में. ओके, बिकॉज ऑफ इट ओनली इट गेट्स ऑरेंज कलर बैंड्स इन DNA. Clear? So by this bands only we can DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as the elution. Elution, are you getting? But I am telling you, आपने क्या करा, DNA fragments को stain करा with which thing? Ethidium bromide. Okay, ethidium bromide with exposed to UV light, so it looks like orange color bands, और आपने उसके बेस पे small fragments को big fragments से अलग कर दिया. Okay, and this step with gel piece, gel के साथ अलग करते हैं. Okay, the DNA fragments purified in this way are used in constructing recombinant DNA. Okay, now your fragments are with you and you can use these desired fragment in your recombinant DNA technology, or you can directly join this DNA fragment in plasmid. आपके पास प्लाज्मिड है, आपको प्लाज्मिड में इसको डालना है, देन यू कैन यूज इट. प्लाज्मिड ऑलरेडी वी हैड डिस्कस्ड. ओके, आई होप दिस थिंग इज क्लियर. हाउ वी कैन सेपरेट आइसोलेटेड डीएनए फ्रैगमेंट. डीएनए फ्रैगमेंट को आइसोलेट करते कैसे, या हमारे जीन ऑफ इंटरेस्ट को कैसे आइसोलेट करते हैं? with the help of different enzyme. Okay, like if we want to dissolve a wall, that time also we required lysozyme. Okay, then protease, lipase. Okay, these kind of enzymes are required to break down cell को पूरा degrade करते हैं बेटा. only DNA, we can't use DNAs डाल देंगे तो DNA टूट जाएगा. ओके, RNAs we use to cut RNA, ओके, प्रोटीन को कट करने के लिए प्रोटीएज, ओके, लिपिड को कट करने के लिए लाइपेज. सो सच काइंड ऑफ एंजाइम वी कैन यूज टू डिसॉल्व होल सेल. ओनली डीएनए रिक्वायर्ड. उस डीएनए के फ्रेगमेंट करके वी कैन सेपरेट देम विद द हेल्प ऑफ जेल इलेक्ट्रोफोरेसिस मेथड. आई होप that much is clear. If you have any query regarding this, please send me your question. Thank you, children.